Hello everyone, in this class we'll learn about immunity. Resistance of body against pathogenic agents is known as immunity. Immunity is of two types. It can be innate immunity or acquired immunity. Innate immunity, which is non-specific immunity. It is the inborn capacity of a body to resist the entry of microorganisms into the body. It represents first line of defense against any pathogen. For example, phagocytosis of bacteria and other invaders by WBC and tissue macrophage system. Destruction of swallowed organisms by acid secreted from stomach and digestive enzymes. Destruction of foreign cells and tumor cells by natural killer cells. Whereas acquired immunity is a specific and it is resistance developed in the body against any specific foreign body like bacteria, viruses, toxins, vaccines. The key to acquired immunity is ability of lymphocytes to produce antibodies that are specific to foreign bodies, that are antigens. Acquired immunity is of two types. It can be active or passive immunity. Active immunity. Acquired immunity developed by individual as a result of antigenic stimulation like previous infections, vaccines. This involves active functioning of the person's immune system leading to the synthesis of antibodies or production of immunologically active cells. Active immunity can be induced naturally or artificially. Natural active immunity results either subclinical or clinical infection, whereas artificial active immunity is by giving vaccines. Passive immunity, acquired immunity developed by an individual through the transfer of blood, serum, antibody from an immune individual in a ready-made form. Here, the recipient's immune system plays no role. It gives immediate response and is transient. It can be natural or artificial. Natural passive immunity, where antibody transmitted from mother to the fetus either through placenta or through breast milk. In artificial passive immunity, resistance passively transmitted to the recipient by administration of antibodies. It is given in the form of serum, for example, anti-tetanus serum. Acquired immunity has two components, humoral immunity and cell-mediated immunity. Lymphocytes. They are responsible for specific immune response. In fetus, lymphocytes develop from bone marrow. The lymphocyte designed to develop the cellular immunity migrates to the thymus gland and are transferred into the T lymphocytes. The lymphocyte designed to develop the humoral immunity are processed in liver during the fetal life and bone marrow after the birth and are transferred to P lymphocytes. There is a third variety of lymphocyte called natural killer cells. The different types of T lymphocytes are helper T cells or inducer cells or CD4 cells or cytotoxic cells. They're also called as killer cells or CD8 cells. Suppressor T cells, they're also called as CD8 cells or T8 cells. Then memory cells. There are different types of B lymphocytes as well, plasma cells and memory cells. Coming to humoral immunity, it is mediated by circulating IgG antibodies in the gamma globulin fraction of the plasma proteins. IgG antibodies are produced by B lymphocytes. B lymphocytes are stimulated directly by foreign organisms or through an antigen presenting cell. Some of B lymphocytes are converted into plasma cells producing immunoglobulin. Each plasma cell continues to produce specific antibodies for several days or weeks till it dies. Some of the B lymphocytes are converted into blast-like cells and they remain as a memory B cells. On exposure to the same antigen subsequently initiate a rapid response due to the presence of memory B cells. Humoral immunity is a major defense against bacterial infection. Immunoglobulins. Immunoglobulins are produced by the plasma cells. It is composed of 4 polypeptide chains linked by disulfide bonds. There are 2 heavy chains which are denoted by H and 2 light chains which are denoted by L. Each light chain lies parallel to the terminal portions of heavy chain. There are two areas, FAB for antigen binding and FC for complement binding. Terminal portions of both heavy and light chains are called variable segment. Specificity of each antibody is due to the specific amino acid pattern of variable antigen. This is the diagram showing antibody. So these are the two heavy chains and these are two light chains, and these are variable regions where it is for antigen binding, and these are the constant regions which are meant for complement and macrophage binding. There are 5 basic types of classes. IgG, it is the most abundant antibody present in our body. It can pass through placental barrier and it provides natural passive immunity. They serve as opsonins and promotes phagocytosis of bacteria. It is the antibody seen in the secondary immune response. IgA is formed by mucosal and submucosal aggregates of lymphoid tissue. From here, it is transported to epithelial cells. This provides effective defense mechanism in mucosa of alimentary canal, lungs, and genitourinary tracts. IgM is the earliest immunoglobulin or IgG to be synthesized. It activates complement system. They are predominantly produced in primary immune response. Antibodies of ABO blood group system belongs to this class. IgG is they mediate allergy, hypersensitivity, and anaphylaxis, chiefly produced in the linings of respiratory and intestinal tract. They release histamine from basophils and mast cells. The last one, IgD, is present on the surface of B lymphocytes and therefore they are involved in antigen recognition. Mechanism of antibody action. The antibody exerts its action by two mechanisms, by direct and indirect. Indirect action mainly is by activation of complement system. Direct action. The antibodies are the multiple antigen sites on invading organisms. So antibodies can activate the invading agent in one of the following mechanisms. They may lead to agglutination, that is, antibodies bind with antigen to form a clump. They may lead to precipitation, that is, molecular complex of antigen and antibodies becomes so large that it is rendered insoluble and it precipitates. They have a neutralization property, that is, antibodies cover toxic sites of antigenic agents and make it non-toxic. They may lead to lysis. Antibodies attack on the cell membrane of foreign organisms and cause destruction of the organism or the lysis. Antibody enhances the opsonization and hence it facilitates the phagocytosis. Some of its indirect actions are it is through the activation of complement system. It is a system of about 20 proteins. Normally these are present in blood and tissue spaces and are inactive. They are activated by antigen-antibody reaction. When antibody binds with the antigen, binding site on antibody for complement becomes activated and that will bind with complements. That will activate the complement system and the activated complement produces certain effects. And these effects are opsonization and phagocytosis, lysis, and agglutination. Opsonization and phagocytosis. Activated complement C3b strongly activate phagocytosis by neutrophils and macrophages. These neutrophils and macrophages phagocytosize foreign antigen or foreign bacteria. Second one, they lead to lysis. One of the most important product of complement cascade is the lytic complex, which is a combination of multiple complement factors C5, C6, C7, C8 and C9. It directly causes lysis of foreign organism. They may lead to agglutination. The complement product changes the surface of invading organism and causes them to adhere to one another or agglutination. They may also lead to neutralization, that is, the activated complements attack the structure of some viruses and make them non-virulent or non-toxic. It leads to inflammatory changes, that is, the increased local blood flow, capillary leakage of proteins into interstitial space, coagulation of these proteins. These inflammatory changes immobilize the organism and prevent movement of organism from the area of invasion to the other parts of the body. So there are warm and cold antibodies. Agglutinogens of EBO system and RH system can react with their corresponding antibodies at a normal or near body temperature, that is in warm environment. Therefore, these agglutinins are called as warm antibodies. But antibodies of some other blood group systems can react with their corresponding agglutinogens only at temperature between 5 degree to 20 degrees Celsius, and they are called as cold antibodies. Coming to cellular immunity, it is produced by T lymphocytes that can directly attack intracellular pathogenic organisms like viruses, fungus, bacteria. They can activate macrophages which phagocytosize pathogenic microorganisms. T lymphocytes acquire an antigen presenting cell for its action. The antigen presenting cells are macrophages, B lymphocytes, dendrite cells in spleen, and Langerhans cells of skin. The major histocompatibility complex 2 is present on the membrane of antigen presenting cell. The MHC, the gene that produces protein MHC1 and MHC2. MHC1 is present on the membrane of all nucleated cells and platelets. T lymphocytes are activated through antigen presenting cell. The antigen presenting cell phagocytoses the foreign organisms, digests it, and separates portion responsible for antigenicity. This gets attached to the MCS2 proteins which protrude from the surface of antigen presenting cells. So this MHC2 antigen complex is recognized by an appropriate T lymphocyte, and T lymphocytes get attached to this complex. This will activate the T lymphocytes, that is the CD4 or helper cells. The activated T cells proliferate and a clone of activated T cell is produced, and some remain as memory T cells. The following effects are produced by the activated T cells. The interleukins are produced and these activate further proliferation of the T cells. Interleukin produced by activated T cells activate B lymphocyte. Activated T lymphocytes activate CD8 or cytotoxic T cells, and cytotoxic T cells cause the cell lysis directly. Activated T cells lead to inflammation and delayed allergic reactions during the second exposure. The memory cells are stimulated and produce more quantity of antibodies at faster rate than the first exposure. Summary of acquired immunity. So when antigen enters the body, it is ingested by macrophages and is partially digested. Antigen peptide fragments then combine with MHC2 and move to the cell surface. This processed antigen with macrophage then binds with the CD4 cell, that is helper cells. CD4 cell is activated and it secretes interleukin 2. Interleukin 2 causes the cell to multiply, forming a clone. The CD4 cells then activate B lymphocytes, causing them to proliferate and transform into memory B cells and plasma cells, or it may activate cytotoxic CD8 cells. The plasma cells secrete large quantities of antibodies into the general circulation, and CD8 can also be activated by forming a synapse with MHC1 antigen presenting cell. So this diagram summarizes the acquired immunity. Okay. So when antigen is digested by the macrophage system, it attaches with the MHC2 complex and it is going to be presented with appropriate CD4 cells, and the CD4 cell recognizes that antigen, and further the T cell it gets activated. The activated T cell may activate the B cells, which converted into plasma cells, and they produce antibodies. Or sometimes, so this is going to lead to the humoral immunity, whereas activated CD4 cells, they secrete interleukin and they further activate cytotoxic T cells or CD8 cells, so which have ability to directly kill the cells. Or some of the activated uh, cells, they they lead to inflammation and delayed hypersensitivity. So sometimes some of the cells, uh, they can get attached with MCH complex, and they uh, can activate CD8 cell and